Mitochondria, the ATP-producing organelles on which virtually every energy-demanding cellular process depends, are also, somewhat paradoxically, the primary generators of reactive oxygen species in the cell. As electrons flow through the electron transport chain during oxidative phosphorylation, a small fraction escape and react with oxygen to produce superoxide and hydrogen peroxide. These RS accumulate in aging mitochondria, progressively damaging the very membranes and proteins the mitochondria need to function, and eventually triggering mitophagy, the selective autophagic removal of dysfunctional mitochondria through the PINK1/Parkin pathway. What vitamin C does in this system is more sophisticated than simple antioxidant protection, and understanding its precise role clarifies why dosing, timing, and form all matter more than popular supplementation culture typically acknowledges. The PINK1/Parkin mitophagy pathway is activated by a specific trigger, the loss of mitochondrial membrane potential that occurs when a mitochondrion becomes sufficiently damaged. This membrane potential loss causes PINK1 to accumulate on the outer mitochondrial membrane, recruiting Parkin and initiating the autophagy receptor cascade. The RS that drive mitochondrial damage are the upstream signal for this entire process. Vitamin C, by scavenging excessive RS in the cytoplasmic environment, reduces the rate at which otherwise healthy mitochondria sustain oxidative damage, effectively extending the functional lifespan of mitochondria that are still performing adequately while allowing the RS accumulation in genuinely damaged mitochondria to proceed to the threshold that triggers their selective removal. This is the Goldilocks function of ascorbic acid in mitophagy. It is not eliminating all RS, which would prevent mitophagy from occurring at all, but buffering the excessive burden that would damage healthy mitochondria and blur the distinction between those warranting removal and those warranting preservation. The precision of this function explains why the post-exercise timing of vitamin C supplementation has become a point of legitimate scientific debate. Reactive oxygen species generated during intense exercise are not merely collateral damage. They are primary signals for the mitochondrial adaptations that make exercise beneficial, including mitochondrial biogenesis and improved stress resistance. Supplementing high-dose vitamin C immediately after exercise appears to blunt these adaptive signals, which is why some exercise physiologists recommend a delay of 2 or more hours before post-exercise vitamin C intake. The body's saturation kinetics for vitamin C favor multiple smaller doses throughout the day over a single large dose. At intakes above approximately 200 to 500 milligrams, plasma concentration plateaus and excess is excreted renally within hours. Split dosing, 200 to 250 milligrams 2 to 3 times daily from food and supplements combined, maintains plasma levels more consistently than a single 1,000 milligram dose that rapidly peaks and declines. Guava leads food sources at approximately 228 milligrams per cup, followed by red bell peppers at 190 milligrams, kiwi at 167 milligrams, oranges at 95 milligrams, and strawberries at 89 milligrams. Raw preparation is preferred because heat rapidly degrades vitamin C. Boiling vegetables can reduce their vitamin C content by 50% or more. Smokers have demonstrably higher vitamin C turnover and require approximately 35 milligrams above standard recommendations, a modest adjustment that population-level guidance underemphasizes given the prevalence of smoking and its connection to accelerated mitochondrial oxidative damage.